Hello, everyone, and welcome to today's webinar, Double Transcriptomic Reads in Single-Cell RNA-seq Using Molecular Depletion. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Jumpcode Genomics. To learn more, visit jumpcodegenomics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Jon Bezney, Research Associate Third, Research and Development at Jumpcode Genomics. Jon, you may now begin your presentation.
Hi everyone, my name is Jon Bezney. I'm a Research Associate 3 here at Jumpcode Genomics, and today I'm going to tell you about one of our products that enhances the signal-to-noise in single-cell RNA-seq. The name of our product is CRISPRclean. If you go on our website, the actual name of this product you'll see here on this slide, CRISPRclean Single-Cell RNA Boost Kit. And like many of you guys know that are familiar with single-cell RNA-seq, while it is incredibly useful at separating out individual cells of different types, it does have its issues. And what I'm showing you here on this slide are some of the issues that essentially confound our downstream computational interpretation. One of those you'll see on the far left panel is that of the total amount and number of mRNA that's inherent within a cell, the traditional single-cell RNA-seq, the 10x methods, you only capture about 10 to 20% of those inherent mRNA. The second part, this middle column, is that the data itself is incredibly sparse. This ends up being an issue for some of our computational processing downstream in terms of efficiency and weeding out if a 0 is an actual 0 or if a 0 is a dropout, aka the mRNA was there but was lost at some point during the experimentation process. And lastly, on the right, that there's a high amount of noise. And what this paper and what this graph is showing you is that the counts per gene of two cells of the exact same cell type, the R-squared value is incredibly low. And you'll see things circled on the far right, like these massive number of dropout events, essentially showing that taking averages across means of cells is not necessarily the best practice in interpreting single-cell RNA-seq.
In order to insert our CRISPRclean workflow into the general 10x single-cell RNA-seq workflow, what you can see on the left is a description of our unwanted reads represented in purple that we remove with a simple CRISPR digestion. And as you can see, we do this before the PCR amplification in order to maximize the number of unique molecules that are recovered. You can see the workflow on the right and the timeline incorporated. In total, it takes about 2 hours from start to finish, but the hands-on time is only 45 minutes.
The outline of this presentation is going to consist of the following. First, I'm going to go over our proof of concept, essentially assess the initial viability of this approach within cardiovascular samples. Second, I'm going to talk about our methodology for designing a robust technology using a mass amount of publicly available data. And third, I'm going to show you the results of our application on immune samples. Now you guys might be thinking, what are we going to be removing within single-cell RNA-seq data? Is there anything useless that can be removed? And so to really set the scene, what I'm describing to you here is essentially a schematic illustrating the reads that go on the sequencer and the reads that get used. So in this graph, on the green on the bottom arrow is the total number of reads. So 100% of these reads, the green reads are what goes on the sequencer. After that, about 90% or so confidently align to the genome and are actually used. Now what you see on the top is this pink bar represents that of those that align confidently to the genome, really only about 50%, 50 to 60% align to the transcriptome. And because this is an RNA-seq experiment, so when you do any of your alignment methods, the most canonical being using Cell Ranger from the 10x platform, then you're really only taking those that align to the transcriptome. And so thus you can see this top pink box highlighted in this pink dash line represents that even if you have 100% of your reads that come off the sequencer, really only about 50 to 60% within this sample example are really being used for downstream processing. So what I'm showing you here is that same schematic, but overlaid on top are the regions and the number of reads that we target for removal. So what you can see here in this light blue box are the non-polyadenylated ribosomal RNA that again are totally removed from the sequencing library, are absolutely ignored. Those account for about 10% of the reads, and thus we target them for removal. In addition, you can see in the dark purple box about 100 of the top highly expressed ribosomal and mitochondrial genes that are typically removed and do not play an important part in identifying unique cell types or cell phenotypes. Those are also removed, and together they constitute about 36% of your reads. So in order to see if removing those regions actually has any effect in redistributing reads to lowly expressed transcripts and unique molecules, and thus increasing the signal to noise, what I'm showing you here is a schematic overview of the initial experiment that we performed. And there's a couple things here to note. One is that we are comparing the depletion protocol and our CRISPRclean protocol to running 10x as is, the standard workflow. And this is incredibly important here because it's essentially, we've designed this experiment to see if our method is valuable above any standard researcher performing the method as is. And the second important thing to note is that each comparison here, while the schematic isn't properly representing it, but each comparison here, we're representing the control versus the depleted, and those two represent the same exact cells. So at the WTA amplification step, after we have cDNA, we essentially split the cDNA equivalently into two groups, one being the control and one being the CRISPR-clean depletion. So essentially each of those groups represents the same exact cells. So computationally at the end when we do our comparison, we're comparing literally the same exact cells, a cell-to-cell basis comparison. And so lastly, as you can see here, based off of the colors, that one of the main questions of this study is looking at endothelial cells within coronary and pulmonary, and essentially trying to answer the question, are there any unique transcriptional differences between coronary and pulmonary across these samples? So if you focus on the graph on the left, the first cohort of samples are the depleted CRISPR-clean samples. And as you can see from the bar graph, you see this is the percent of reads that essentially align to the transcriptome and end up being used. So you can see right off the bat from these base metrics, we get about a 20% boost in the amount of reads that are used in downstream processing. Now if you focus on the right, you can see at a sequencing depth about twofold greater than the standard sequencing depth, about 25,000 or 30,000 reads per cell. You can see the added value of performing depletion. We recover about 250 more genes per cell. We recover an additional 2,000 unique molecules per cell. And because of that, what we see is about an additional 800 or 900 genes that are incorporated into downstream processing and thus are biologically relevant. Now what I didn't tell you about these samples is that these are only endothelial samples. So they're really predominantly only one main cell type, and the goal of this analysis is to really dissect individual subtypes within this one main cell type. So what you can see here represented in these UMAP plots, that in both the standard and depleted, there's only really one cohort of cells with many different subtypes That cohort representing again, one main endothelial type is represented in this purple dashed line. Another thing that's fantastic to see is that the depleted doesn't alter the underlying transcriptional state of these cells, represented here is that the UMAPs are almost a mirror image of each other. Now what's very interesting, and the added value of depletion becomes apparent once we begin looking a little bit more closely at some of these subtypes within these endothelial cells. So what I'm doing here is the next couple of slides, I'm essentially picking randomly some of these subtypes within the main cohort of cells and looking at the differentially expressed gene relative to all other subtypes within the sample. That's being represented here within these volcano plots. And you can see on the left in the standard, if you're looking at this as a normal researcher, you would look at this volcano plot, and you would see cohorts of genes both upregulated and downregulated past the log2 fold change of 2 to 4. So you would look at this and you would say, this is a massively different subtype. The cells represented in this orange sample are wildly different from the cells in its surrounding neighborhood. Now, if you look on the depleted, remember we've added an additional close to 250-300 genes per cell, but more importantly, we've added an additional 2,000 unique molecules per cell. These samples here represent about 50,000 cells. And so by redistributing those reads to lowly expressed and essentially reducing the dropout and noise within the sample, what you can see here is a greater biological interpretation that represents the ground truth. The same number of genes are different amongst this same exact cell type. The only difference is a massive shrinking in those thresholds, in those log2 fold change. So if you look at this as a researcher now, you don't see those massive differences in genes that you saw in the standard condition. This again, like I said, more accurately represents the ground truth, that this is one main cell type, and these are just different nuanced subtypes within this main group of endothelial cells. I'm showing you here a different subtype within the group of endothelial cells, just to show that this is pervasive across all subtypes. It's not just one subtype. And essentially by redistributing these reads, we do a fantastic job of collapsing the differences that were initially just inherent noise within the data. You can imagine, again, that in the standard condition, if we have genes that are only captured by those cells simply because they were dropped out in the sequencing data within the other cells, that they essentially end up being interpreted as phenotypic signal. By removing those and redistributing reads more evenly across lowly expressed and capturing more unique molecules, we can see a reduction in this technical noise, and we do a better job of interpreting the biological data. Now you might remember that one of the main goals of this was to be able to discern transcriptional differences between coronary and pulmonary. Now, what you're seeing here are the results of batch correction, following the same exact batch correction, the same exact computational methods. On the left, when you have, again, batch correction, 50% of these cells are coronary, 50% of these cells are pulmonary. What I'm highlighting in the purple dashed lines is that massive regions where the batch correction essentially does an incredibly poor job of overlaying cells of these different types and creating one integrated manifold interpretation. Now you can see that on the right, the coronary and pulmonary, essentially every single colored subgroup within that graph is represented in both coronary and pulmonary incredibly evenly. It's almost as if they're mirror images of each other, except for only that one region that I've highlighted in purple down below. Again, this intuitively makes sense. These are endothelial cells from blood vessels. The only difference, so they're the exact same cells, the only difference is where those blood vessels are located. So when we perform a differential expression analysis between the coronary and pulmonary cells, what I'm showing you here are the results of the top 8 genes that come out of that analysis. So the first 4, represented in the x-axis on the top, are genes that are essentially upregulated within coronary pulmonary, within coronary, excuse me. And then the next 4 are the top 4 genes that are downregulated in coronary relative to pulmonary. As you can see, the same genes are showing up from the analysis. So we're not losing any of this biological signal. But you can see, if we wanted to pinpoint which of these subgroups is really the differentiating factor, lending to this difference between coronary and pulmonary cells, if you focused on the heat matrix on the left within the standard condition, it would be incredibly hard to pinpoint any one of these subgroups as the main causal factor kind of driving this difference between coronary and pulmonary. But by removing that technical noise and adding an additional 2,000 unique molecules per cell across 50,000 cells, you can see it becomes incredibly clear that there's really only one subgroup that is driving this difference between coronary and pulmonary endothelial cells. Now, just to confirm that our findings are on track with previously published datasets in bulk analysis, what I'm showing you here is a network diagram, the gene set enrichment on the left of that one subgroup. And you can see highlighted in purple are the genes that are playing the most significant part in these biological processes that you can see on the bottom right. Things like blood vessel morphogenesis, external encapsulating structure organization, and extracellular matrix organization. Now, what's interesting is that all of these genes have been previously identified in bulk analysis, leading to the buildup of plaque within coronary blood vessels. So this essentially shows that while depletion increases this biological interpretation, that it's not something incredibly nuanced, that it makes sense based off of previous published data sets and bulk analysis. So we did a fantastic job of removing about 36% of unwanted reads and redistributing those, leading to an increase in about 2,000 unique molecules per cell. But are there any other reads within our data that can be removed that are unnecessary to interpret any sort of biology? So what I'm showing you here is the exact same schematic that I showed at the beginning. The light blue representing the non-polyadenylated ribosomal reads, the dark purple representing the polyadenylated ribosomal and mitochondrial reads. And now we're adding 2 extra groups that we have targeted for depletion. One represented in kind of this turquoise right next to the ribosomal RNA reads is what's labeled as unaligned reads. And unaligned reads essentially are these consensus intervals across all sample types and tissue types that are these genomic intervals that essentially show up across all of these data sets. Perhaps they are poly A stretches that get captured and amplified during the RT generation. And second, represented in the light purple, next to the dark purple ribo-mito, are these non-variable genes. And you can think of these as highly expressed housekeeping genes whose expression does not change across cell types. So essentially, if it doesn't change across cell types, then it plays no role in interpreting and distinguishing different cell types within single-cell RNA-seq data and is perfect for removal. So what we did to ensure that this was a robust group of genes and regions targeted for removal, we essentially performed in silico depletion of 14 publicly available single-cell SRA datasets. So on the far left, if we only removed the non-polyadenylated ribosomal RNA, that represents about 10% of aligned reads. In dark purple, if we only removed the top expressed ribosomal and mitochondrial genes, represents about 30% of reads. And remember, these data points is across all 14 sample and tissue types. If we combine those two together, we get up to about 42%. Now, if we add on, in addition to those guide sets, if we add on the guides targeting those unaligned genomic intervals, we can get up to about 50% of reads that could be removed and redistributed to lowly expressed, rare, and unique molecules. And lastly, if we add on those non-variable genes, the genes that aren't playing any role in biological interpretation, we can get up to about 58–60% of our reads that could be removed and redistributed. So the schematic I'm showing you here is the experimental overview of what we performed combining all of these guide sets within PBMC samples. So what we're showing you is on the far left is that we're comparing our depleted conditions that were performed in triplicate against the control. And the control here, again, is just the standard 10x workflow. And same as before, each of these control versus depleted condition represents WTA splits of the exact same cell types. So right off the bat, if we look at the distribution of reads in the far left on the control, you can see in green that represents not informative transcriptional reads. In blue represents informative transcriptional reads, and in purple represents the rest of the reads that align to the genome and thus aren't used. Now on the right, what you can see is a almost twofold enhancement in that blue region, or in other words, a 100% increase in the reads that map to the transcriptome that are informative. Now, if we dive in a little bit further and look on a cell-by-cell basis, you can see on the far left a sequencing saturation curve that as we increase in sequencing depth per cell, we're simply recovering more and more unique molecules per cell. At the end of the day, at a sequencing depth of about 25,000 reads per cell, depletion recovers an additional 280 genes per cell, and about additional 500 unique molecules per cell. And this correlates to an additional 2,000 genes that contribute to biological signal that otherwise would not have been found within the control. Now, before I jump into some of the UMAP plots, for those of you that are familiar with the analysis of single-cell RNA-seq, I want to ensure everyone that I am not and did not perform any manual annotation of this dataset. In order to automatically identify the cell types within PBMC databases, we leverage a tool that uses predefined legacy markers. So this is an automatic annotation tool that essentially references massive repositories of single-cell PBMC data containing over 1 million cells. So when we use that tool on both data sites, you can see here some of the canonical cell types that show up within PBMC cells. Those included on the far left, like pink, are showing up, these natural killer cells. And on the right, those same natural killer cells are now in the bottom right corner identified in blue. So you can see we're recovering these same exact cell types with one notable difference. On the left, kind of highlighted and circled in this purple dashed line, you can see that the control is unable to distinguish between these CD4 monocytes and these two kind of arms that are branching out from the CD14 monocytes. Whereas in the depleted condition, you can see one of those arms actually separate within transcriptional space and identify and distinguish between CD14 and CD16 monocytes. In addition, it also has the ability to characterize these rare cell types of dendritic cells, CD1c positive dendritic cells, that represent less than 1% of the cell population in absolute frequency. Just to confirm that the results previously mentioned were not a factor of the computational analysis or automatic annotation tool, what we did is we performed this unsupervised cell clustering that uses deep learning to automatically identify the proper number of clusters represented within your data. Using this tool, what we found is that depletion still recovers 2 additional cell types using this unsupervised clustering approach. On the far left, you can see labeled the control sample, we find about 9 clusters, and in the depleted sample, we find about 11 clusters. You can see on the bottom left in the control, cluster number 6, the depletion, because of the redistribution of genes and the additional 2,000 genes that are playing a part in biological interpretation, cluster number 6 is split into 2 individual clusters. And lastly, this kind of arm that connects cluster 1 and cluster 0 in the control is essentially totally separated into its own cluster, and you can see there's no longer any sort of connection between cluster 0 and cluster 1. In addition, to give us some confidence in assuring that these two additional clusters are actually real, this tool also assigns a probability of those clusters being separate clusters. The probability you can see as a colored heat matrix on the right of this graph, going from about 0.2 being yellow all the way up to 0.8, 0.9 represented in dark blue, dark purple. So you can see those additional clusters in the bottom right that were separated, as well as those in the top right in the depleted. This tool assigns a very high confidence, a high probability, assuring us that these are actually two different clusters. So what I've showed you up to this point is that we can sequence the same and recover more information by performing depletion, but on the other end of that coin, can we sequence less, perhaps half the number of sequencing reads, but recover the same exact information using CRISPRclean depletion? So what I'm showing you here are the raw metrics within this table. This first column represents the control, or the non-depleted. So at a sequencing depth, about 27,000 reads per cell. You can see the same number of cells in that second row. But on the bottom two rows, genes per cell, we recover about 1,300 genes per cell and about close to 2,000 unique molecules per cell. Now the second column represents if we just took half, the same exact sample, and sequenced to half. You can see here again representing about 13,500 reads per cell. We don't recover exactly half, but we recover significantly less genes per cell and UMIs per cell in those bottom two rows. Now, if we take essentially that same exact sample of cells, perform depletion, and only sequence to half, you can see we recover the number of genes per cell and the number of unique molecules per cell. And on the right, what I'm showing you is a distribution of those reads and genes per cell and UMI counts per cell of the depletion at half the sequencing depth and the control at the full sequencing depth. You can see there is no bias in the distribution of the data and almost a perfect overlay of the counts per cell and genes per cell. Even more surprisingly, now when we compare the UMAP plots of the control at 27,000 reads per cell and the depleted at half the sequencing depth, in the depleted condition, we surprisingly still recover the ability to identify those two additional cell types that were unable to be identified within the control sample. So just to recap, I would like to point out that we recover an additional essentially 100% boost in informative reads within our PBMC samples. We recover an additional about 1.5-fold increase in genes per cell and unique molecules recovered per cell. And this enables us to recover an additional 2 cell types within our PBMC sample data that could not have been identified otherwise. So all in all, depletion increases the sensitivity of single-cell experiments and allows one to recover the same information at half the number of sequencing reads. So in conclusion, depletion recovers more information per cell. We increase the signal to noise by performing depletion. And by redistributing those reads, depletion statistically enhances the biological interpretation. I would like to thank the entire team here at Jumpcode Genomics, as well as the Ponde Lab at Scripps Research, as well as the Scripps Genomics Score that helped with the sequencing. This was a team effort and could not have been done alone. And please let me know if you have any questions.
Thank you, Jon, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, does the CRISPR-Clean reaction use all the cDNA from my 10x prep?
Hi, yes, that's a great question. So the answer is no, it doesn't. So the traditional 10x workflow is after you generate the oil droplets with your cells, go through and generate the cDNA, or the WTA is what they call it, the whole transcriptome amplification. At that point, you come to a nice stopping point. And at that point, what you can do is you can quantify your cDNA, and you actually split your cDNA into 4 equivalent groups, even according to the protocol. So what you do here is in depletion is you just take one of those splits and perform the depletion on one of those splits. So if you want to compare it to a control, like you saw at this presentation with our 2 experiments, then you essentially only use 2 out of the 4 splits. One would be your control, your standard 10x processing, and then the other one would be for your depletion, go through the protocol, which also raises another excellent point that I just want to reiterate, is that all of the experiments that you had seen, because we're splitting it at this cDNA, the control and the depleted represent the same exact cells. So the comparisons here are incredibly robust, as if you're comparing, if you had 10,000 cells, it's almost as if you're comparing every cell against itself.
Okay, thank you. Next question. Does CRISPR-Clean depletion introduce bias to my gene expression readout?
No, absolutely not. So this is one of the first things that we looked for. And so if you take every gene in a control sample and every gene in a depleted sample, and if you look at simply the correlation or the R² value of all 15 or 17,000 genes that you have, it would be incredibly close to 1. It's all the samples we've seen are 0.98, 0.99, so essentially round up to 1. So the key here is that we're not changing the distribution of the genes or changing the relative expression. We're simply providing kind of more value for what you would get, where you take the reads that are typically totally ignored and removed computationally, and we're taking those reads and redistributing them to lowly expressed genes and unique molecules that otherwise wouldn't be captured. So we're just increasing the sensitivity and specificity without biasing the gene expression.
Okay, thank you. Next question. Where can I find detailed information on this kit?
Yeah, absolutely. So if you go to jumpcodegenomics.com and you can browse through our website there, you will see some very much detailed information about some of the specifics on this kit. And if you remember at the beginning of this presentation, look for CRISPRclean SC Boost, and you'll be able to find more information about the experiments that we reviewed today, some of the detailed metrics, as well as some of the actual raw data. If you want to peruse through the data yourself and kind of check out that we see this added boost in unique molecules and genes, and you can also download the information on our website as well.
Great. Well, it looks like we have time for one more question. Is this kit compatible with the 5' chromium assay? What about Visium spatial?
Yeah, so that's a great question. So one of the reasons, if you remember about the middle of this presentation, that we did a deep dive into accessing publicly available data, is we wanted to make a product that was not only universal, really across different biological samples and different replicates, and as well as different tissue types, but we wanted to make sure that the reads that we were seeing were also found in other kits and other assays. And so we have now done some preliminary experiments within a 5' chromium assay, and it worked fantastic, as well as some preliminary experiments within the Visium spatial, and we see very similar effects where we're able to remove these unwanted, unusable reads and redistribute them to lowly expressed reads. So yes, it seems like this is pervasive across a lot of platforms within 10x Genomics.
Great. Thank you again, Jon, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Jumpcode Genomics, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.